on to this last session of botany, the last score session for botany here. So all of you must have enrolled in the score test. So just last few days left. If you have not yet enrolled, then do enroll for a score and get a chance to evaluate your talent and compete with some of the best mind in India. So with the score, you will not only get the opportunity to show your talent, but you will also get an idea where do you stand in this current competitive environment. So in this, along with that score exam, we have also started some very interesting session which will help you in doing good in score. So this will be the last session for botany, where I will discuss some very important MCQ, or you can say, you can consider this as the mock for the score test, right? So let's start with this. So here initial questions will be from morphology. So we have already discussed a lot about cell cycle. So cell cycle this time I have kept in the end so that we can focus more on morphology in the beginning. So here first question from the morphology, the root system, right? So there are two types of root system, tap root system, and if we say here there are two types. So there are two types of root here, tap root system and just a second. Yeah, so tap root system and fibrous system and adventitious root system. So tap root system, they have a primary root, right? Then they have a secondary and tertiary root. So these are called as the lateral root. So there is a primary root and there is a lateral root. So let's see. Tap root system, it originates from radicle, correct? It is a typical of monocot plant. Tap root system is found in dicot. It is a characteristic of dicot, not monocot. Monocot have fibrous root. So this is wrong. It produces lateral root of several order. Yes, it produces lateral root of several order. So this is correct. So both 1 and 3 are correct. So 1 and 3 is correct. So C will be the answer here. Let's move ahead to the next question. So here, if we go to the next question here, this one. Conquering region of the root, which of the following are current? Now, there are regions of the root. It is region of meristem, region of elongation and region of maturation. So based on the regions of the root, it asks these questions. So first, root cap protects the apex. Root cap protects the apex as the root pushes through the soil. So this is correct. Root cap pushes the root inside the soil. So this is a correct answer. So here this one is correct. Regions of elongation consist of actively dividing cells. Region of elongation consists of actively, no, it is consist by region of meristem. Region of maturation bear root hair. Region of maturation bear root hair. Meristematic region is few millimeter above the root. This is also correct. So first, third and fourth is correct. Second one is wrong. Now, then if we further move ahead. Now, which of the following statement about root modification are correct? Root modification. Carrot and turnip are example of adventitious root modified for storage. Adventitious root? No. Carrot and turnip are modification of tap root. So first statement is wrong. Prop root support the banyan tree. Yes, you have seen banyan tree, prop root is coming out. So second is also correct. Stilt root arises from lower node in maize and sugarcane. Again it is correct. And pneumatophore in rhizophora help in gaseous exchange. So yes, It is also correct. So except one, all are correct. So correct answer will be 2, 3 and 4. So here 2, 3 and 4 is correct answer except one. What is wrong here? Carrot and turnip, they have taproot. They are taproot. So they are modifications of taproot. Now let's move ahead. Root cap protects the delicate apex of the root. Root cap protects the delicate apex of the root. Correct? It is thin, delicate structure that absorb water and mineral. Right? It is a thin, delicate structure that absorb water and mineral. Is it root cap which absorb water and mineral, or is it root hair? So assertion is true, reason is false. So we will go with C. A, assertion is true, reason is false. Further, this question. In sugarcane, supporting root emerge from the lower node of stem. Yes, lower node, sugarcane. These supporting root are known as prop root. No, they are called as tilt root. Maize and sugarcane, tilt root. Prop root is present in banyan tree. In monocot plant, the primary root is short-lived. Monocot plant has fibrous root. So primary root is short-lived. Correct statement. It is replaced by large number of root arising from the base of the stem. This is also correct, right? So primary root is short-lived and replacing it is the number of root arising from the base. So assertion and reason are both correct. But is the explanation correct? If I say, so for that what we connect? We connect by because monocot plant, the primary root are short-lived. Because, no, so this explanation is not correct. So it will be not a correct explanation. Next, the region of elongation in root is responsible for increasing the region of elongation in the root is responsible for increasing the length of the root. Yes, elongate, increase the length of the root. The cell in this region are very small and continuously divide. Continuously divide, not region of elongation, it is region of meristematic. So assertion is true, reason is false. Carrot and turnip has adventitious root. Carrot and turnip have adventitious root. Is it correct? Carrot and turnip have tap root, not adventitious root. Their root are modified for storage of food. This is correct. So reason is correct, but assertion is false. So D, assertion is false, reason is true. Match the type of root with their examples. Now here, if we say tap root system. So tap root will be with, tap root generally is present in dicot stem, plants, right? So here which is a dicot? See, banyan and gram both are dicot, right? But banyan will go with prop root, so tap root will go with gram. So tap root, gram. Fibrous root, you can go with maize, or fibrous root can be sugarcane. Stilt root can also be sugarcane and maize. Prop root will go with banyan. So here I will match it for you so that you can clearly see here. See, so tap root will go with tap root. So tap root will go with gram. Fibrous root, let's consider this as maize. Then prop root, banyan. Then stilt root, sugarcane. Right? This is the correct match. Then next one. Potato is a stem modification. Root is done. Now we are talking about stem. Potato is a stem modification. Yes, because it has node, internode, right? It bear node, internode, buds and scales of leaf. Correct? So assertion is correct, reason is correct, and reason is the correct explanation of assertion. Match the stem modification with their example. First, rhizome. So rhizome will go and match with rhizome. So rhizome here is ginger, tuber, potato, bulb, onion, sucker, mint. So this is the correct explanation. These are the stem modifications. Then moving ahead, Which of the following statement is true about stem tendril? Stem tendril. So stem tendril arises from the axillary bud, right? Yes, stem tendril form from axillary bud. They help the plant in climbing, again correct. And they are seen in cucumber and watermelon. They are seen in cucumber and water. See, there are two types of tendril. One is called stem tendril, another is called leaf tendril. Leaf tendril is found in pea and stem tendril is found in cucumber and watermelon, which are called gourds and grapevine. So 1, 2 and 3 all are correct. So D is the right answer. Then potato is underground stem. Potato is underground stem. Yes. Sweet potato is root modification. Correct. Sweet potato is which root modification? Adventitious root modification. Onion is bulb and underground stem modification. Yes, onion is bulb, underground stem modification. Carrot is a stem modification. Carrot is also a stem modification. It is a root modification. We have seen carrot and turnip root modification, but taproot modification. So this is wrong. Rest 1, 2, 3 are correct. So 1, 2 and 3 is correct. Fourth is wrong. Further, identify the correct statement about underground stem modification. Rhizome has nodes and internode. Yes, rhizome has nodes and internode. Tuber show axillary buds. Tuber show axillary bud. Potato, you all see eye in the potato, they are axillary bud. Bulbs like onion have fleshy scale leaves. Yes, onion, what the edible part of onion is actually the leaf. So they have fleshy leaves. Tap root of carrot is an underground stem. Tap root is not underground stem, it is a root. So 1, 2, 3 will be the correct answer here. It is a flattened, now it is regarding the phylloclade. What is phylloclade? When the leaf become fleshy and green and the stem and the leaf modify into thorn, right? Cactus you see, that is the phylloclade. So it is a flattened cylindrical green stem, correct? It takes over the function of photosynthesis. Found in xerophytes like opuntia and cactus, correct? And it is a leaf, it is not a leaf modification, it is a stem modification. So 1, 2 and 3 is the right answer. Which of the following statement is correct for stilt root of maize and sugarcane? They are adventitious root. Yes. They arise from node of the stem. They arise from the node of the stem, right? So when we say stilt root, so they arise from the node of the stem. Yes. They provide mechanical support. Yes. And they are modification of stem. They are modification of stem. Stilt root is a root. They are not stem modification. So again 1, 2 and 3 is correct. Fourth one is wrong. Which of the following statement are correct about thorn? Right? Now here stem thorn. In leaf we call it spine. So if we say thorn, so the first statement here, right? Stem modification. So yes, thorn is a stem modification. It arises from axillary buds and they perform defensive function, right? They form defensive function. This is also correct. And they are present in bougainvillea and citrus. This is also correct. So all of them are correct here. Now moving ahead, next, if I say, so now, so this was all about the morphology. So mainly I have covered the section root and stem. Similarly, go back, revise the leaf part, flower part, other parts also. Now coming to the cell cycle, very important part, cell cycle. During which phase of the cell cycle are DNA and histone protein synthesized? Where is DNA and histone protein synthesized? So for that, let's see the point. We know that DNA and histone are synthesized during interphase, right? We know that interphase has 3 stages: G1 phase, S phase and G2 phase, right? So DNA and histone are synthesized in S phase, right? So let's see here, they are synthesized in S phase. Then next, here the centromere divides during when the centromere divides. So centromere will divide, remember this, if this is the chromosome, right, and this is the centromere. So this centromere will split during which case? It goes this side, this goes this side. So they split during anaphase. The centromere split during anaphase. So it is split during anaphase 1 of mitosis, right? It is split during anaphase 1 of mitosis, and it also split during anaphase 2 of meiosis. So here you can go with B also and you can go with D also. Then next one, which of the following is feature unique to meiosis and not mitosis? Means difference between mitosis and meiosis. First, chromosome condensation. It happens in both the case, meiosis as well as mitosis. Crossing over. Now crossing over is unique to meiosis. There is no crossing over during mitosis, right? Here you will see crossing over, and that's why due to crossing over there is a variation that is not taking place during mitosis. Next, synapsis. The pairing of homologous chromosome occur during. Synapsis is pairing of homologous chromosome. When does it take place? So here prophase has, you know that prophase has how many stages? Prophase has 5 stages, right? Liptotin, zygotine, pachytine, diplotin, dikinesis, right? Liptotin, zygotine, pachytine, diplotin, dikinesis. Now here every stage has certain characteristic through which we identify those stages. So here synapsis, right? So synapsis is the characteristic of zygotine. Along with synapsis, if bivalent is asked, if tetrad is asked, right, in all the case, or synaptonymal complex is asked, all are formed here in this stage. If crossing over is asked, so crossing over occur in pachytine stage. Is it clear? So here the correct answer will be synapsis will occur in the zygotine of prophase 1. Another question, a bit interesting question. If a cell has 46 chromosome, right? Let's consider this is a cell and this has 46 chromosome. 46 chromosome. In which phase? It is in the G1 phase, right? It has 46 chromosome at the G1 phase. Fine. Then it is asking that what will be the chromosome number after S phase? After G1 phase, what happened? In S phase, the DNA replication take place. But does that replication double the DNA, number of DNA? It doubles the DNA content, the number of DNA or the number of chromosome, not number of DNA, number of chromosome remain the same. So remember, the DNA content get double, but the number of chromosome remain the same. So here 46 will remain 46. Remember here, if I say in G1 phase, S phase, G2 phase, right? If in G1 phase the number of chromosome is 2n and the DNA content is 2c, then in S phase what will happen? The number of chromosome remain the same, but the DNA content become double and it will continue in the G2 phase. So what you can see in G1 phase and S phase, the number of chromosome remain the same. It's the DNA content which get doubled. Is it clear? So 46 chromosome will remain 46 in the G1 phase. Then there comes a Anaphase. There comes Anaphase. In Anaphase, what happened? The chromosome number, we say number of chromosome double. Number of chromosome get doubled. If question is asked from you that in which stage the number of chromosome get doubled, you will say Anaphase. And rest of the point. Now, this will be common stage for mitosis and meiosis, G1 phase, S phase and G2 phase, right? But after that in meiosis it will change. Clear? So these are some of the point you have to keep it in mind, and from here a lot of question is asked. Now to the next question. In mitosis, the number of chromosome in daughter cell, in mitosis, the number of chromosome in daughter cell is, so mitosis, what we see? In mitosis, what we see? If there is a cell, then mitosis, the number of cell will get doubled, right? One cell will become 2 cell. If the DNA content is 2N and 2C, so here what will happen? The number of chromosome will remain the same, right? Number of chromosome remain the same and DNA content also remain the same. So is there any change in number of chromosome in mitosis? No change in number of chromosome. So no change in number of chromosome and also no variation. No variation. No variation. So in mitosis, the number of chromosome in daughter cell is same as the parent cell. This is parent, daughter. So it will remain the same. Then which of the following statement is correct regarding meiosis? Which of the following statement is correct regarding meiosis? First, 2 division but DNA replication occur twice. Now here let's understand this. Just a moment. If I say here, let me explain it and then I will tell you the answer. First, let's understand the meiosis. In meiosis, what happened? One cell will become 2 cell, right? And then these 2 cell will further become 2 more cell. This is how they divide, right? Means one cell become 4 cell. This you can call it as meiosis 1 and this one is meiosis 2, right? So how many division is here? So there are 2 division, but replication how many? 1. So we say that there are 2 division but 1 replication. So first 2 cell division but DNA replication occurred twice. No, DNA replication once, division twice. 2 division but DNA replication occur once. So yes. So that's why we say 2 division and 1 replication. 2 division and 1 replication. Is it clear? So this you remember, in mitosis what happened? 1 division, 1 replication. And in meiosis, 1 division, 2 replication. Now further moving ahead. In which stage of prophase 1 does crossing over actually occurs? Remember, leptotene, zygotene, pachytene, diplotin, dikinesis, right? So what we have seen there in the leptotene, right? First the condensation starts. This is very common question which is asked. That's why it is repeated. Now zygotine, as I have said, synapsis formation, bivalent formation, tetrad formation, and synaptonymal complex formation. Then after zygotine, pachytene, here crossing over take place, which is the question. Then diplotene, here what will happen? Here we say chiasmata formation. Chiasmata formation, which is an X-shaped structure, right? Which is an X-shaped structure. And then finally in dikinesis, terminalization. So remember here, if question is asked terminalization, you will say dikinesis. Chiasmata formation, diplotene, right? Then crossing over, zygotine, synapsis, bivalent, tetrad, or formation of synaptonymal complex, you will say zygotine. And condensation of chromosome starts in, you will say leptotene. Another question can be arrange them in correct order. They will give you some random order. So remember, leptotene, zygotine, pachytene, diplotene, and dikinesis, right? So that's the way you remember it. Then comes next, colchicine arrest mitotic division at which stage? For that, let's first understand what is colchicine. So colchicine is a chemical which is also called as mitotic poison. Colchicine is called mitotic poison. Why we say it adds mitotic poison? Because it arrests mitosis. Arrest mitosis. Why it arrests mitosis? Because it prevent spindle formation, or in this is a very loose term, spindle formation. Actually spindle formation just remain as it is, right? Usually spindle should elongate, it should contract, all those change in the spindle fiber is blocked, and due to which the chromosome will not be able to segregate. And in which stage the arrest take place? This happen in the mitosis, and that is why it is called arrest, mitotic arrest, right? In mitosis it happen, and of course in which phase? Metaphase. Because after metaphase what happened? In metaphase the spindle fiber is formed, but in anaphase they will segregate. Now in metaphase only if you arrest the spindle fiber, will anaphase take place? No. Chromosome will segregate? No. So cell division will stop, and because it has a tendency to stop the cell division, colchicine is used as a cancer drug. Colchicine is used as a cancer drug, because you know that in case of colchicine what happened? There is in case of cancer what happened? Uncontrolled cell division. And when there is uncontrolled cell division, then somehow we need to stop the cell division, and for that we need colchicine which control the cell division. So cancer is also controlled. That's why it is also used as a cancer medicine. So these were some of the important questions for score. Now I think we have already covered a lot of questions from all the possible topics and all the possible chapter important questions. So I hope that this will help you in doing good in score. So I expect all of you to give your best for the score. Best of luck, and I hope that you all will do great in score. So best of luck, and lastly, don't forget to enroll In score, just few days left. Tell your friends, family, relative, neighbor to get enrolled in score because only few days are left. Your small decision can bring a lot of difference in your life. So best of luck and have a nice time.